So a lot of people are joining. That's exciting to see how many people are joining. So we wait for, I think, another minute to ensure everybody is coming out of the lobby into this exciting seminar. Yeah, Paul is already laughing. It's good to hear you, Paul.
Thank you, Janine.
All these antibody engineers, antibody therapeutics people interested in learning more about antibodies. It's great that they're all joining, and we are so fortunate, Paul, to have you. I'm still seeing people joining, so let's wait another few seconds, and then we're getting started. Do you also hear the beeps, Paul?
Yeah, it's kind of like
It is always funny. And at a certain moment, we should get started because it's getting, you could say, it's a pity if you lose time on waiting for people. So let's get going. So dear antibody enthusiasts, and I mean with enthusiast, everybody who really enjoys to learn more about antibodies, is working on antibodies, or whatever, feel welcome. The Antibody Society welcomes you to this very special seminar. And you could say it's a celebration seminar because our beloved therapeutic modality, and you can see it here in gold on the cover of the October issue of Nature Reviews Immunology, this antibody molecule turned 50 this year. Of course, we can debate about birth date, but let's not do that. Let's go on the date we use as the birth date. That's the 7th of August, 1975, when George Köhler and Köhler and Milstein invented hybridoma technology, or invented, they published it at that date. And we take that as the birth date. Paul Carter will take us along the journey of our favorite therapeutic modality. He recently published a very nice review article about our molecule in Nature Reviews Immunology. You see the cover on this slide. And he will take us along the opportunities as well, the history, the opportunities also with respect to the future. Before I introduce the speaker of today, Paul Carter, some logistics. A lot of people are joining, and we still hear this beeping in the back. At least Paul and I are hearing that. But it also brings us for a challenge. There will be some time for questioning, but we can't let everybody speak up in this seminar because we are here with more than 500 people. There is a Q&A button in the chat, so you can post your questions in the Q&A button, but in the Q&A chat, and it would be super helpful if you put your name and also the organization with that. We hope we have enough time to moderate the questioning, and if your question can't be answered, we apologize in advance. Furthermore, the presentation will be recorded. All the participants in this call will get an automatic link, but it will also be published on the YouTube channel of the Antibody Society. Now, Paul Carter. Paul Carter, I think everybody needs, no issue. So you might not need that much of an introduction. There is so much to say, but I think I still want to do some highlights. You're in the biotech world for over 40 years. Today, you are a Genentech fellow working in the antibody and engineering department. But before this, you have been working at Genentech before, also Immunex, Amgen, Seattle Genetics. And I think also nice to say that you are very tightly linked to the Antibody Society. You've been part of the board of directors, and you have also been a president for the Antibody Society. What I think is very relevant to mention here, you are a visionary. And I think that also makes you being such, I would say, a role model for all of us. You're a visionary, you're an inventor, and just a few things where you have been visionary and been inventive. You've invented antibody humanization methodologies, the bispecific antibody technology, which everybody is now using because it's having freedom to operate. So you were far ahead of the time. The knobs into holes technology. You're an inventor of a common light chain technology, and far more other things. But I think most of all, I personally see you as a scientist by heart. You've authored so many impactful publications. You've delivered so many always enjoyable and insightful keynote presentations and other presentations. And of course, shared sessions and built conference programs. And Paul, I will never forget our time when we were chairing the Santa Fe Keystone Conference, when we were thinking about COVID, which hit right after that. But most of all, you're extraordinary kind. You're collaborative. You're fun. And you're so involved in the antibody community and a role model for all of us. So without further ado, Paul, the floor is yours.
Well, thank you, Janine, for that really just extremely kind intro. And it's, I'm delighted to reciprocate. I've enjoyed working with you so much over the years. So it's a great honor and privilege for me today to present this webinar. And truly we're here to celebrate a monumental accomplishment in the field. That's the invention of hybridoma technology depicted in this cartoon, which of course we're all familiar with. And this really was the launching point for the whole field of antibody therapeutics. It would be hard to overstate the impact of monoclonal antibodies. We're now over 200 approved antibody therapeutics that really have become ubiquitous as research reagents and great sources of intellectual property. And of course, the potential of monoclonals was correctly recognized by the Köhler and Milstein being awarded both the Lasker Prize and the Nobel Prize in 1984. This picture is taken from the party at the MRC Laboratory of Molecular Biology in 1985. So we see Cesar and another laureate, Max Perut. And so what I'm going to do today is share some of their the journey of monoclonals in an article that I published with a couple of my colleagues, Andy Chan and Greg Martin. And this was really a kind of a labor of love writing this review. At the time, in 1984, I was a graduate student with Greg Winter, and I was in the room, although I think I was camera shy and not in any of the photos that I've been able to find. So here we are as a field now. So, with our starting point with hybridoma in 1975, 50 years later, we're now over 200 antibody therapeutics and impacted many different disease areas, particularly oncology, autoimmunity, and inflammatory diseases. Of course, this is a huge and mature field, and so both with the review and preparing this webinar, it's challenging to decide what to focus on. So thanks everybody for coming today, and I've chosen a few things listed here to highlight, which I hope you'll find interesting and enjoyable. So firstly, starting with, well, what were the key steps that really helped us as a field to make antibodies relevant as therapeutics? How the landscape has evolved, focusing particularly on bispecifics and antibody-drug conjugates and routes of administration. I'm going to illustrate some progress in the field with advances in antibody technologies and how they've improved patient outcomes, focusing on targeting HER2 in oncology and targeting B cells, which has been a great medical value in oncology, also autoimmunity and inflammation. I'll comment very briefly at the end on future opportunities and just have a brief closing remarks. So if we look back over the last 50 years, I'm kind of struck just what a long and winding road it was to develop antibody therapeutics, starting with the hybridoma technology in 75. 11 years later, the first approval. It really wasn't until the 2000s that antibody antibodies really started to take off as therapeutics, and you know, as mentioned, we're now well above 200 approved antibody therapeutics. So, you know, what did this take? And of course, there was many things. First off, basic research and understanding of disease, understanding targets, generating therapeutic hypotheses, how you could intervene in disease with antibodies. Lots of preclinical testing, antibodies for safety and efficacy, and then clinical trials. I mean, it quickly became apparent that there were some major challenges with mouse antibodies in terms of their clinical potential. And so as a field, that was one of the major challenges to understand and address these challenges. I think perhaps it's sort of an unsung hero in the development of antibody therapeutics is advances in biotechnology innovation, and particularly being able to make antibodies on a huge scale, from kilogram to multi-ton, and really pristine pharmaceutical grade material. So going back to the late 1970s and to early 80s, it quickly became apparent with early testing of mouse monoclonal antibodies that they had some shortcomings as therapeutic agents. Notably, these three, that they're foreign proteins, which elicited an anti-drug response in most patients. They're typically short plasma half-life in humans, which limited their target exposure, and they're inefficient effector functions. And so as many of the early targets were killing cancer cells, this was a limitation. Over the ensuing years, actually, even decades, it became sort of appreciated how important the human Fc was in terms of giving the possibility of long plasma half-life and efficient effector functions. As for overcoming immunogenicity, really the first steps in this direction were domain swapping, so swapping their mouse constant domains with human constant counterparts to create chimeric antibodies, and then the humanization pioneered by Sir Greg Winter in humanized antibodies. Of course, in parallel with that, there were major advances in developing human antibodies through transgenic animals, phage display, and later yeast display, and cloning from B cells. And together, these are engineering initiatives, technology initiatives have largely overcome the original known limitations of mouse antibody in terms of reducing the immunogenicity, being able to achieve longer plasma half-life in human, and also efficient effector functions as of where this is needed. So if one looks at the technologies, so this is, again, looking at all of the antibodies which have been approved to date, excluding biosimilars. But this time, rather than looking at therapeutic area, we're looking at the technology that was used to generate them. And what I'm struck by is that really this is dominated by humanized antibodies and human antibodies, with murine and chimeric playing a much more minor role. So let's come back to the our unsung hero of our field, and that's biomanufacturing. So over a period of decades, starting with the birth of biotech in the mid-1970s, this has been, you know, scaled from 2,000 to roughly 25,000 liter in stainless steel tanks. And really the CHO cell lines have been sort of the workhorse for antibodies, with also myeloma cell lines featuring prominently. This has enabled the large scale production of pharmaceutical grade antibodies. Another important facet, which was highlighted in this beautiful review from Brian Kelly, was the advances in the titer by combining improvements in cell line development and also optimization of cell culture. So, you know, over really this period of several decades, we've gone from less than 1 to about 2 grams per liter, all the way up to fairly routinely, 8+ grams per liter. I'm looking forward at sort of current innovation in biomanufacturing. This includes the use of single-use technologies up to about 2,000 liters, and this provides both flexibility for capacity, because it doesn't require the same cleaning in place for stainless steel, that it saves on water and energy. Another major innovation has been continuous bioprocessing with automation, real-time sort of monitoring for faster production. So let's turn now to some of the evolution of the field, and particularly bispecifics and antibody-drug conjugates, or ADCs, and also routes of administration. Here is the same collection of 200 or so approved antibodies, but this time we're considering these approved antibodies from the perspective of what technology that they use. As you can see in this large blue segment, that's IgGs have really dominated the field for a long time. But it's also kind of clear that when looked over the last 10 years, particularly the last 5 years, just kind of the exciting emergence of different variations on the antibody theme, particularly antibody-drug conjugates and bispecific antibodies that are focused on today. I think a particularly exciting area is the emergence of the living drugs, so the cell-based therapeutics, such as CAR T cells, which are cell-based therapy but utilize antibody fragments for targeting. So I'd like to kind of just offer a perspective on the history of bispecifics, and it's stunning, really, just how long this has taken, and just how many difficult problems had to be solved along the way. So they're one of the pioneers in our field of bispecifics was Alfred Nessenov. And so the first paper that I'm aware of with bispecifics goes all the way back to 1960. It took until the 2000s with the approval of 𝘁𝗵𝗶𝗀𝗶, the first bispecific, and then blinatumomab, and so I would say, from my perspective, is that it was really the success of blinatumomab that really excited and reinvigorated the field with our sort of bispecifics. And so what did it take? Well, it took the first 25 years to come up with a broadly applicable application of bispecific, so particularly T-cell engagers. So that was a seminal paper from Michael Bevan in 1985. And bispecifics, as you guys all know, are complex molecules, and can be very hard to make, particularly on a gram to kilogram scale for the clinic, and making them really pristine. And so there are a couple of technologies which were enabling for these early approvals, the so-called hybridoma, and then for blinatumomab, so the BITE format. So these enabling technologies were truly sort of pioneering and help advance the field. And they also had their challenges. So if you look at the hybridoma, which involves co-expressing 2 different antibodies in the same cell, in addition to the desired bispecific, one can get up to 9 additional unwanted chain pairings, which reflect mispairing of heavy and light chains, and so heavy chain homodimerization in addition to the desired heterodimerization. And this is a kind of a challenge that sort of resonates with me, because I remember hearing Köhler and Milstein present this technology back in the early 80s when I was sort of a graduate student, not knowing that it would sort of feature quite prominently in my own career, and of course many others to try to address this challenge. As we look back on this, several years later, I think there's thanks to the ingenuity of many in the field, we actually have some great solutions for being able to make bispecific IgGs efficiently in single host cells. And I've kind of divided them broadly here into technologies to overcome light chain, heavy chain mispairing, and ones to force the heterodimer or overcome the homodimerization of heavy chains. And so, you know, there are more technologies available now than I could respectably fit on one slide, but I think it's kind of exciting to see where we've come as a field. And we now look at the rise of bispecific antibodies as therapeutics. Really, this was a long time in the coming, and it's really quite stunning, the progress since 2020. We're now up to at least 20 approved bispecific, including this sort of blue segment, which is sort of bispecific IgGs. There are other formats here, including fragments, 2+1, 2+2 bispecifics, biparatopics. And so this is a cartoon of the T cell engager concept. And so you can see with the approved bispecifics, to date, this has been dominated by the sort of the T cell engagers. So as we think about the future of bispecifics, I think there's understandably a huge amount of effort across the field to try to develop improved T cell engagers, including trying to enhance safety, therapeutic index by activity on demand. And here are some of the emerging technologies. So masking antigen binding, pH-dependent binding, and the ATP switch technology from Chugai. In trying to enhance tumor to normal selectivity, one thing which springs to mind here are the 2+1 bispecifics and avidity engineering, or the use of trispecifics. You know, as a field, we've been very successful with T-cell engagers with hematologic cancers. Solid tumors is proving to be a difficult nut to crack, probably for many reasons, including access and T-cell exhaustion. And so there's a huge amount of activity, including the use of co-stim, either combos of bispecifics or trispecifics. I think another exciting area with bispecifics are emerging applications, and sort of one obvious thing to consider here are the brain penetrant bispecifics. This is a graphic to illustrate that from a recent review article, and of course many groups have used bispecifics that target the transferrin receptor as a way to get a molecule across the blood-brain barrier. Other innovations, including the sort of combining checkpoint inhibitors and anti-angiogenics, and also using bispecifics in the context of antibody-drug conjugates. So turning now to ADCs, and this is also kind of just a time of great innovation and progress in the field. And you know, when we look from, say, the last 10 years, so 2015, 2020, 2025, it's kind of really just amazing growth in the number of approved bispecifics. And what we were kind of struck by when we were writing this review is just the proliferation of different payloads. So, you know, it started with the 𝘁𝗵𝗶𝗀𝗀 for double-stranded DNA breaks. We've got the auristatins and the maytansinoid as tubulin polymerization inhibitors. We've got the camptothecins as TOP1 inhibitors, and then the PDB dimers for DNA cross-linking. And you know, in thinking about sort of ADCs, kind of realized that there's a lot of decisions that one has to make in designing ADCs. and make good, if not great decisions for many different things to be successful. And so these including the target antigen. Ideally, this should be efficiently internalized with an ADC, restricted expression beyond target cells. Of course, one has the delivery vehicle. Typically, this has been an IgG, but although other things have been tried, sometimes it's engineered for site-specific drug conjugation, including engineered cysteines, non-natural amino acids, conjugation through to carbohydrates. Ideally, this is efficiently internalized as an ADC. There are lots of choices to be made with the linkers and the payload. So with the linker, the goal is to achieve stability in circulation and then release in the tumor. So this could be that the linker itself is cleavable, or it could be uncleavable, but the payload released by catabolism of the IgG. As for the payload, typically these are ultra potent drugs. So-called drug antibody ratio, that's the loading of the drug on the antibody. It's typically in the range of 2 to 8. The conjugation, as I mentioned, the different technologies, often this is through cysteine and lysine, although there are other methods too, and then stable in the circulation. So there are, in thinking about the ADC field, there are still many areas of innovation which make me very optimistic about the future of the field. There's one of what I would consider as the grand challenge with ADCs is trying to reduce off-target tox from ADC catabolism, and this is illustrated in a graphic from a nice review. These are 6 ADCs which are approved for solid tumor targets, and in the box, and I'm sorry, this is a little too small to probably to project well other toxicities. And so, the reality is most of your ADC ends up somewhere other than the tumor, and when your ADC is catabolized, that's a potential source of tox. So this is sort of one of the grand challenges in the field to try to address this. Other major areas of innovation include the dual cytotoxic payloads to try to overcome drug resistance, and some of these have advanced into clinical trials. I think a particularly exciting area is the innovation in looking at different payloads, so not just beyond cytotoxics, immunomodulators, protein degraders, glues, antibiotics, and nucleic acids. We have different delivery vehicles, both different forms of antibodies, antibody fragments, non-antibodies, and something which I've mentioned previously is sort of activity on demand, and this may help with on-target off-tumor tox. There's still room for improvement with drug-like properties. So if your payload is a very hydrophobic molecule, maybe by choosing your linker carefully, hydrophilic linker, or the environment, how you conjugate it to the antibody, you can improve the drug-like properties. I want to switch now briefly to delivery, and so most antibodies are administered IV, that's this blue segment, or subcutaneously by injection. There are a few which can be administered either IV or subcutaneous, and so one doesn't have to look very hard to really appreciate why subcutaneous delivery is so appealing. In contrast to IV, this can be performed in minutes rather than hours. Self-administration at home is possible, including some pre-filled syringes. And so this can really be beneficial to patients in terms of convenience, compliance with improved quality of life, and lower healthcare costs. But subcutaneous delivery comes with some significant challenges. One is limited to small volumes, and that often necessitates high dosing concentration. I would say a big challenge in this area is so-called bioavailability, and that's to say the fraction of your antibody that gets from the injection site into circulation, and this can vary over twofold. Understanding antibody high concentration properties is still very much a work in progress, and for some antibodies this can lead to high viscosity aggregation or insufficient solubility. In this graphic, and kind of illustrating some of the benchmarking the sort of manufacturing limit, which is about 30 centipoise, against some everyday liquids. So the manufacturing limit is roughly the same as vegetable oil, and then the subcutaneous injection limit is roughly 12 centipoise, which is about the viscosity or slightly more than the viscosity of blood. Now, there are a number of ways that one can enhance the potential of antibodies for sub-Q delivery. Today, I'm just going to focus on one of these, which is highlighted, and I'll come back to a little later in the talk with the HER2 antibodies, and that's being able to deliver larger volumes by co-formulating with hyaluronidase, which will digest the connective tissue and allow larger volumes to be delivered safely and comfortably. So now I'm going to move into giving you some examples where these technologies have played out, and I'm going to start with some examples in HER2, which we've worked on at Genentech for many years, and so are familiar with. We're also going to touch on B cells targeting. Of course, the field is absolutely enormous, and it wouldn't be it's not possible for me in the review we wrote, or certainly in one presentation, to do justice. So please bear with me. We've tried to pick some examples which are reflect some concepts rather than try to cover everything that's been done by us collectively as a field. So firstly, a very brief background to HER2. It's a member of the EGF receptor family of growth factor receptors. It's an orphan, which is to say there's no known sort of ligand. So the four family members are represented in the cartoon. So HER2 can form homodimers. HER2 can also form heterodimers with other family members, and you can see it's quite complex in terms of the different ligands for EGFR, HER3, and HER4. Really, one of the key and seminal steps in the field was a collaboration between Dennis Lehman at UCLA and Axel Ulrich when he was at Genentech, and so in this very productive collaboration, they discovered that HER2 is overexpressed in about 15 to 20% of breast cancers, and that the HER2 gene amplification is correlated with the poor prognosis in breast cancer, as illustrated in this 𝘁𝗵𝗶𝗀𝗀 paper from this groundbreaking publication they had in 1987. So this really kind of excited our interest at Genentech, and I think more broadly in the field. Let me show you first a kind of an overview of some of the sort of major antibody therapeutics targeting HER2. So let's start on the left. This is a cartoon of HER2 showing the four different domains. So targeting the domain 4, which is the membrane proximal domain, we have trastuzumab, which I'll share in a minute. There's effector function enhanced related antibody called margetuximab. So these will support ADCC and also have anti-proliferative activity. We have perpetual that binds to a different domain, domain 2, and this blocks heterodimerization with HER3. In the middle of our screen here, we have their ADCs, and I'll come back to those in a couple of slides' time. And then we also have a biparatopic, which targets the domains 4 and 2, so kind of combine some of the attributes of trastuzumab and pertuzumab. So let's kind of reflect on some of the early days with HER2 with antibodies. So colleagues here at Genentech and Brian Friendly developed a panel of anti-HER2 antibodies, and we took one of these antibodies 45 and humanized it. Gail Lewis and her lab demonstrated that this humanized antibody had potent ADC activity, and here we're looking at killing of a number of different cell lines which have varying levels of HER2 expression. So you can see, at least for this panel, with more HER2, you get more efficient killing. And then so Genentech in collaboration with Jeff Ravich's lab showed that humanized antibody had anti-tumor activity in mouse. And then clinical trials with the humanized 45, which went on to become trastuzumab, started in mid 1992. Of course, at that time we waited at bated breath. We had no idea whether we had done anything useful and whether the humanized antibody would actually have any benefit to patients. So the reality was certainly exceeded my sort of wildest expectations, and I'm going to show you a couple of their early clinical trials showing the benefit of HER2, and in this first one with Dennis Lehman as the first author, showing that when Herceptin was added to chemotherapy, that it gave a progression-free survival benefit. And then a few years later, in an earlier disease setting, see that when Herceptin was added to chemotherapy, that it actually gave a significant increase in disease-free survival. Of course, this inspired us and others to try to do even better. And one of those initiatives at Genentech was led by Gail Lewis, Mark Slakowski, and others. And what they did was develop an antibody drug conjugates. It was based on trastuzumab. It uses the மெட்டான்சinoid DM1 as a payload conjugated through lysines. A DAR of approximately 3.5. So thioether linkage, so non-cleavable, and so we believe that the mechanism of release is catabolism of the antibody. The metabolites are charged, so it likely has no bystander killing activity. So this, we're kind of delighted to see that when you compared to small molecule combo, the trastuzumab ematanzin was outperformed the small molecules, and then in a direct comparison, there was the trastuzumab ematanzin, also known as TDM1, outperformed the parent antibody. So that was very exciting. And of course, many other groups have innovated in this space, and I think one of the ones which has particularly excited those of us who've worked on HER2 is this molecule. It's the trastuzumab deruxtecan. It was developed by Daiichi Sankyo and AstraZeneca. So again, it uses trastuzumab as a delivery vehicle. It uses a different payload, so camptothecin, a topo-1 inhibitor. It's a higher DAR. It's about 8. It's cleavable linker. Cleavable by cathepsins, and it has bystander killing. And here showing you some of the clinical data that were published a few years ago showing that this ADC outperforms the physician's choice of chemotherapy. And something that I want to point out here for those of us who worked on HER2, which is really quite stunning and extremely impressive, is that what we're looking at here is activity against HER2 low breast cancer. And that's been really incredibly difficult to treat with antibodies targeting HER2. So hats off to everybody who's involved in developing this AC. It's kind of a major step forward in the field. So let's kind of quickly wrap up this section by kind of commenting on where we are in targeting HER2. So here's a summary of the monospecifics. I've talked about trastuzumab. Pertuzumab is another antibody. This one blocks the heterodimization with HER3. We have some effector function enhanced antibodies. We've got the bioparatopic that I mentioned earlier. We've also got a bispecific HER2, HER3. I've already talked about their ADCs. I think another area of excitement with their HER2 targeting is to co-formulate with hyaluronidase. And as I alluded to earlier, this allows one to dose larger volumes, which means that one can transition from IV administration to sub-Q. So now I'm going to turn to targeting B cells in disease, particularly oncology, autoimmunity, and inflammation. When you're writing this review, we're kind of just astounded just how many antibodies had been approved. At least at that time, which is about 6 months ago, we were up to, as a field, 37 approved antibodies targeting different B cell lineages and 9 different antigens, which are listed here. And so you might be wondering why different groups have gone after different antigens. And I think that's kind of reflected by looking at the development of their sort of the B cell sort of lineage and our B cells at different stages express different antigens. So you can see some of the ones I've just flagged here with the blue asterisks are some of the ones which have been targeted with antibodies. So by targeting different markers, you can target different stages. And so, for example, if in multiple myeloma, the targets which have been pursued successfully include CD38, BCMA, SLAMF7, and GPRC5D. And one other additional benefit is that one can potentially switch from, say, one antigen to another if you have patients that don't express the antigen or they become resistant by down regulating that antigen. Here's one of the first anti-CD20 antibody to be approved, was rituximab, the chimeric anti-CD20, and this was one of the first, if not the first, randomized studies to be published comparing a chemotherapy regimen known as CHOP with adding rituximab to that, and it's really just really an impressive improvement in event-free survival, which is shown here, but also included in this paper was response rate and overall survival. And if one kind of fast forward to the present day, it's really kind of astounding how far the field has come in terms of different approved chimerics, humanized and human antibodies, including several which have enhanced effector functions and beyond oncology, which was the original focus, B-cell diseases and autoimmunity and inflammation. And so in the case of rituximab is used in both settings. Payload and antibodies have also featured a lot with their targeting B cells, and really kind of the pioneers in this area were the radioimmunoconjugates and the immunotoxin. they were not without their challenges, and were ultimately both in terms of safety, kind of sort of logistics and other considerations, and ultimately withdrawn from the market. But they were kind of important stepping stones in the field, and I think now we have a collection of different antibody drug conjugates, which have really just been very successful in B-cell targeting oncology. This has also been a very fertile ground for the T-cell engagers, starting really with the pioneering blinatumomab that really excited so much interest and activity in the field. Many different bispecifics targeting both different antigens, and we have different formats, including the common light chain, 1+1, 2+1. This has also been kind of a very exciting area in terms of the advent of CAR T cells, and this is a really these are delivering drugs. They're having a major impact in oncology, and earlier in clinical development are also being explored for autoimmunity and inflammation in targeting B cells. So kind of wait with bated breath to see how that plays out in the future. So to kind of wrap up this section, and to kind of summarize where we've come as a field with targeting B cells, we have at least 37 approved antibodies targeting diverse B cell lineage targets. We've got really just an amazing collection of different formats with different mechanisms of action. There are way too many indications to list on the slide. And so what I've done is just to say, what are some of the major indications, both in oncology and also autoimmunity and inflammation. So now to kind of wrap this up, I have I'm going to comment very briefly on future antibody therapeutics, and then just a few closing remarks. So thinking about the future of antibody therapeutics, one doesn't have to look very hard to realize just how many different opportunities there are. Of course, like many others of our science, other areas of life, AI is starting to have a major impact in sort of antibody design. I think it's both in starting to happen in de novo antibody design. I think an important application is in multi-parameter optimization. That's to say, to try to improve several different properties simultaneously, because this is hard to do by conventional methods. There's plenty of opportunity to enhance or enable delivery. We've talked about brain delivery earlier. There is progress in the field in oral delivery. That's still a very hard thing to do. Intracellular still seems like it's a long way off, at least with antibody proteins, but perhaps closer with our delivery of antibody genes for expression inside cells in a therapeutically relevant way. Another area of innovation where there's already a lot of progress in the field is developing more robust routes to agonist antibodies. As we get more creative as a field, more adventurous, we also have to consider the immunogenicity risk, and I think there's still a need for better tools to understand this risk and mitigate it. I've alluded to at different times they're overcoming resistance, either because you've got loss of antigen or some other mechanism. In oncology, I think we're very good at as a field at making astonishingly potent molecules, including C-cell engagers and ADCs. It's still tricky to do this and to maintain acceptable safety. One can take IgGs and try to give them properties of other isotypes which are very different, or maybe engineer these other isotypes directly as therapeutics. There's lots of opportunity for optimizing antibodies for sub-Q delivery, which I've sort of mentioned today. And then let me close by just reflecting on what we're here to honor today, which is the monumental achievements of Köhler and Milstein. They closed their paper in 1975 with a comment that such culture could be valuable for medical and industrial use. And of course, that has been incredibly well demonstrated over the last 50 years. And that reflects many generations of our researchers, basic, translational, clinical, and biotechnologists. And of course, intrepid patients and their families who took the risk to therapies that were still at an experimental stage. It's kind of look forward to the future. There's been such incredible progress across the field, and as I tried to highlight very briefly in the previous slide, there's still plenty of opportunity, and so this leaves me very optimistic about the future of our field, and I can't wait to see what happens next. So with that, I thank you for the opportunity to present to you today. I hope that thanks everybody for coming, and I hope that some of this was interesting to you. So thank you.
Thank you very much, Paul. And you see all these clapping hands. I hope you can see it as well, Paul. Okay. It looks fantastic, and hearts, and keep on pushing that buttons, people. It's really nice to see that happening. Paul, thanks so much for taking us through the highlights of our favorite molecule, the antibody, which is a fantastic therapeutic modality, and also through the maturation, you could say, the diversification of the field. And it keeps on going. The hearts are getting more intense, Paul. We got some questions already in the chat, so I think we're going to open the Q&A.
Okay, let me. I think you're going to read them out, and let me see if I can. I think I should be able to read them too. Is that right? And yes, I got it. Yeah, okay.
I think so. But I will read them out loud because I think that is very interesting for the whole discussion. There's two people actually asking a kind of similar question, and that is Sashi Yantiani and Anna Ritter, who are asking a question around the Fc backbone for antibody-drug conjugates. Should it be fully silent? And what would be the effect of a fully silent or an active Fc on PK/PD production efficacy? Sashi states the jury is still out, but maybe you can be the jury here, Paul.
I would say I don't know the answer to that. I mean, certainly when I was at Seattle Genetics in the early 2000s, we got sort of very, interested in that area, and it was kind of very hotly debated at that time whether you wanted something silenced or whether, if you ramped up the effector functions, you could get, multiple mechanisms of action. And I would say, to be honest, I haven't followed the field, really closely enough to know exactly where we are as a field. But those are the kind of discussions that we were having at that time. So as far as I know, the jury is still out. If there's somebody on the call who knows more up to date than me, please chime in the chat.
Yes, that would be fantastic. If someone wants to make a statement in the chat around this and wants to play for the jury, that's very much appreciated. Yeah. The next question is from Marie Cosco-Villebois. Great to see you here in the call, Marie. I'm going to read out this question out loud, and it comes back to the intracellular targeting, which is one of the challenges you already mentioned in your presentation, Paul. What do you think is the probability that the field will be able to target specific cell types, for example, neurons? And then it goes in towards in vivo intracellular antibody fragments that bind to pathological proteins, which are the key drivers for specific diseases like tau, alpha-synuclein, TDP43.
I think that's a wonderful question, and I think it still seems like a very hard problem for their future. It seems like what you really need to in order to be able to, you need to achieve a bunch of different things. If you really want to reach specific neurons, well, you've got to get your virus or whatever delivery vehicle sort of into the brain. You've got to get selectivity for cell types, perhaps by using, biotrispecifics or some kind of, and not gating. And then you've got to deliver your fragments. So, I mean, it may happen in the future, but it certainly does. It seems like it's a lot of big challenges is there. But I love the questions. Thank you.
Yeah, so it's definitely you are also saying that needs a lot of attention from us as antibody drug discoverers, Paul. Is that correct?
No, yeah, no, I think that's right. I mean, I think, if I would say, one thing which kind of struck me in writing this review is just, how many incredibly difficult problems were to solve. And, when we kind of, started as a field 50 years ago, there were problems that we started to understand in terms of limitations of mouse antibodies, but there are other problems that you don't even know that you don't know until you get into the clinic. So I think part of their intent of writing this review was not just to and also putting this together the presentation is say, okay, here are some of the hard problems that as a field that we faced to hopefully kind of attract interest and attention. It's not to say that I'm smart enough to know exactly how you would do that.
No, if that would be the case, then I think the problem would have been solved. Another question comes on AI and machine learning. So that comes from 𝘁𝗵𝗶𝗀𝗶𝗀. So what are your thoughts on the use of AI and machine learning in antibody discovery and development? And the next question is, how promising is it?
I think that's a great question, and I think one of the things that we've been wrestling with here at Genentech, and of course everybody across the field, is to say try to pass out the hype from the reality, figure out what's problems are important to solve and what are kind of tractable with AI, because you either have large data sets or can generate them, or you can iteratively, the so-called lab in the loop, generate some data. And so I think that's a work in progress. There are things which personally appeal to me. One of them I alluded to was the multi-parameter optimization. So let's say you want to increase your binding affinity, and simultaneously, there are some kind of bioactivity, have a pristine developability profile, favorable high concentration properties. If you could figure out how to do that with AI, or just even just design, use AI to figure out how to make antibodies, favorable high concentration properties, that would be a big deal. Or if you could use AI to really better understand immunogenicity risk and engineer that out, those would be sort of huge accomplishments. So I think that is part of our challenge as a field at the moment, is to broadly explore AI and figure out where it's useful, but just do that in a curious but critical way, not just kind of buy into the hype. But yes, yeah.
I think you made a nice statement. Don't buy into the hype, but it is promising, and in particularly from the practical perspective. But be cautious and critical.
Yeah, no, I think that's right. Explore, have fun, but also you need to know when what you're getting out from AI is complete nonsense, and when it's actually useful.
So it needs still wet lab experiments.
Yes, I think I'm only kind of a big fan of that, so-called lab in the loop, where you both, combine AI and test things, experimentally.
Thanks. Then we have another question from Richard Jones. Isn't one of the main barriers in monoclonal antibody development today the expectation that candidates must have a strong single agent activity? Many mechanisms are inherently combination dependent, and under the current standards, even drugs like Herceptin, whose full benefit emerged only in combination, might never have been advanced. So Paul, this is your vision with respect to single agent activity, and of course, in particularly for smaller biotechs, the huge challenge to develop drugs.
I think that's a wonderful question. I think it's very insightful. I don't think I have, frankly, a great answer to that. I mean, I think that's absolutely true. I think in particularly in their, cancer immunotherapy space, there you could just expect from, theoretical grounds that it's that you're not going to get single agent activity or don't expect to see it. And so that is our. I don't think there are easy answers to that, but I think it's an important problem to focus on more.
And maybe, Paul, that's something there where maybe the Antibody Society could make a stand or something in the future. So maybe we should make a note and think about that as Antibody Society.
Yeah, that sounds good, Janine.
Yeah, someone on the line who takes the note. Anyway, next question from Jing Zhao. How can we make antibody mimic more precisely? So make, for instance, cell interactions in the immunosynapse more precise, more precise.
Yeah, I'm not sure that I quite understand the question. I mean, I mean, if you're say, with the T cell engagers, of course, there are many different, bispecific formats that have been explored, from IgGs to fragments, choice of their epitope, whether it's kind of closed membrane proximal or not. So my understanding is that as a field, we've been typically more successful with membrane proximal epitope for T cell engagers, but I think there's still kind of empiricism for exploring different formats for T cell engagers, and just trying and seeing what works best. I mean, I think hopefully as a field we'll get more sophisticated as a time to be able to predict that. But I think some humility here to say we don't understand all the rules, or even if there are rules, and just be willing to try some different things.
So maybe that's also something where you could say, if we curate our data sets better, where AI can be of greater help. What do you think, Paul?
Yeah, no, I think that's a big deal. I think there, data quality, data curation, and also being able to capture things which didn't work as well as things that did, so that because you need both the successes and the things that didn't work to be able to train.
So Paul, I like that. So actually you're now arguing we should curate better data sets for all our AI methodologies, including negative data. And then maybe we can also hit both the technical challenges, but also maybe biological challenges we face. I think that might be a nice, a nice segue to the next question. It's from Yuan Lei. He asked, we've been struggling to expand the therapeutic window for immunotherapies. What directions should the field of antibody engineering explore to potentially solve this therapeutic window problem?
I think that's a great question, and again, I would say, I'm not a magician. I don't have the answers here, but I would say the things that appeal to me, I'm kind of excited by the progress that's been made in the field in terms of exploring activity on demand. So things which are conditionally active. It seems like there are theoretical reasons for thinking, well, that might actually be helpful in terms of therapeutic window. And I think over the next, there's a lot of work which has been done in that area, and I think as a field over the next few years we'll get a better understanding of how useful or not that is.
And Paul, what do you think? Because if you look at the target space we are sampling, and also the two examples you included in your presentation, HER2 and CD20 or B cell targeting, we're still circling around, you could say, relatively, in particular in the solid cancer space, unselective targets. Is that also a way to expand?
I think potentially. I mean, I think with HER2, I think there's the kind of avidity engineering strategies for the trying to preferentially target higher levels of HER2, overexpression and spare their normal tissue with low levels of HER2 expression. But there's also kind of like a not easy, because you'd also like to be able to target, as with one of the ADC examples that I mentioned, to target HER2 low. So it's tricky. You can of course, just say go for dual targeting with our, different combinations of targets, but that's tricky too, because you could then potentially get escape by loss of one or the other of the antibodies. So I think I think what this discussion is highlighting, and it's just some really kind of meaty challenges for us to to tackle as a field, and really just to be able to home in on what we kind of collectively feel are the really kind of critical challenges and opportunities for the next generation of antibodies. So yeah, stir the pot a bit in that direction. Then I would say this talk was successful from my perspective.
Thanks so much, Paul. We are running out of time, and we are already six minutes past the full hour.
Yeah, I think we're in extra innings.
Yes, and there are questions still popping in. So I'm sorry we can't go to all the questions, but I will just pick one last question. And apologies for all the people who put it in questions and didn't get the attention. But I pick a question which got 4 thumbs. So that's what I'm doing here. It's 𝘁𝗵𝗶𝗀𝗃 who asked the question: do you think display techniques such as phage display are likely to play an increasing role in antibody discovery?
So I would say, let me kind of parse that question slightly and say, well, I mean, first off, say, well, in terms of display technology, we've got phage and yeast and other methods. I would say, there are lots of good ways of making antibodies. One of there are things which really appeal to me about some of the display technologies of being able to select for, say, binding to multiple things, or binding to one thing but not another. So to me that's an incredibly powerful capability, and I think there will be an ongoing use for that kind of sort of capability. Whether they will increase more or not, that's a harder thing to say. But I think there's absolutely a place for selection technologies where you can exquisitely select for what you bind and what you don't bind.
Would that be a better way also to chime in all these data sets from, you could say, we need to curate better to take, you could say, learnings?
I mean, just saying, I'm sorry, I'm not sure I understand the question, Janine. Do you mean from just in general, or just with phage, with the display technologies?
Yeah, the idea I have, if you would use display technologies, phage display technologies, you might be capable to generate more data quickly.
I think that's true. Yeah, I think with the display and kind of their NGS, they are kind of powerful ways of being able to generate sort of their very large data sets quickly, which I think might kind of tie in with their the AI theme from earlier.
But the multi-parameter might be lost then with respect to production yields and other, you could say, key elements why the antibody molecule is our favorite therapeutic modality. And there are still questions popping in, and I'm so sorry, but I think we need to close this session. Again, a huge virtual applause for Paul for giving this fantastic presentation, but also for taking so good care of all the questions. Also, a huge thank you to all the participants. Without you, this would not have happened. So thanks for participating, active listening, and asking questions. I also like to invite you all to stay in contact with the Antibody Society when we are at conferences, or reach out, whatever you want to do, and maybe bring forward topics you also are very interested to learn more about. And maybe, no promises, we can follow up on these topics in seminars, education materials, or maybe when we meet at conferences or other places in the world. So feel free to reach out, and for now, have a very nice rest of the day or evening or whatever, wherever you are in the world. Thanks so much.
Great. Thank you, Janine. Thanks everybody for coming. Thank you. Bye-bye. Bye-bye.